eukaryote DNA replication. Now, as I mentioned, the units of the polymerases are very similar. They do about the same job. It's just that in the eukaryotes, you have a more elaborated system for the replication. So the first thing, there is a difference between the eukaryotes and the prokaryotes replication. Remember, with the eukaryotes, you need to have that RNA primase that's going to make that little primer so that the DNA polymerase III can go in there and continue the synthesis. Now, with the eukaryotes, you don't really have that. Everything is in one place here. You have the DNA polymerase. Those are not 1, 2, 3s. Those are the Greek letters instead of the numbers. So now you have polymerase alpha. That this polymerase alpha has several subunits within itself. So one part of this subunit is actually going to add the primer. So now you get the primer out of this polymerase instead of waiting for another molecule to go there and synthesize a little primer. So you get the primer, polymerase alpha is going to get there and work as a primase. It's going to put a little primer in there, especially at the Okazaki fragments, because remember, with the leading strand, you just put the primer once, and then you get the other DNA, that polymerase that is going to continue synthesizing. That's no problem. The lagging strand is always what we have to think about. So now, okay, polymerase alpha going there with the primase is going to add the primer to the leading and the lagging strand. And now you're going to have to switch between this alpha, because you don't want to make a DNA-RNA hybrid, you want to make only DNA. So that alpha subunit has very low attachment to that DNA, is going to fall off. It has low affinity. So when it falls off, you have another polymerase here, which is a polymerase delta, that will grab onto the end of that primer and keep on polymerizing, and it's going to keep on making your DNA. Now, the polymerase delta will replace the alpha and just finish the job, and it has also high processivity. It has 3' to 5' endonuclease activity. Now remember, it's synthesizing how? 5' to 3'. Right? Now, why do you think it has 3' to 5'? That's the reverse. It's the same idea as DNA polymerase III. As I was explaining before, when you have the DNA polymerase synthesizing its DNA, eventually it's going to make a mistake. Okay, it's not perfect. But then when it makes the mistake, it will realize that there is a wrong base in there. So in order to correct the mistake, it will have to install a little bit, go backwards. Backwards means 3 to 5, not 5 to 3, right? So go backward, and it cuts out the wrong piece of DNA, and then keeps on going forward. It just resynthesizes the whole thing, hopefully without the mistake this time. So when you have the switching between the polymerase alpha, the delta is on the lagging strand, you have this process that's called polymerase switching.
C is for clue, that's okay with me.
Which is very easy to remember. Why do you need polymerase switching? Because if you keep the alpha, you're going to have a DNA-RNA hybrid. Now you want to have only the primer for RNA there, and then you keep on going with the DNA. So that's why you have to switch the polymerases. Now, it's not very clear whether the epsilon is the main polymerase for the leading strand, and then the delta would be for the lagging strand. But this is the main idea. One of them is going to be more active in one strand than in the other. They do the same job. Okay, just say polymerase switching from alpha to one of the other two. Another difference, when I mentioned before, you have about 6 DNA polymerases, I thought it was 5 or 6 now, that are present in the prokaryotic system. This is still a small amount, but then on the DNA polymerases of the eukaryotic systems, you have about 13. Where is this? That work in different ways. You have other DNA polymerases that are going to work in the mitochondria, but this is only to make you aware that not the whole system, not the whole cell is using the same DNA polymerases. You don't need to know about the replication of DNA in the mitochondria. Okay? Just keep in mind that when you inherit those polymerases, you're only going to inherit it from the mother, because the mitochondria is only going to come from the egg, right? Because the sperm doesn't have much of it. Another thing that has to happen with the DNA in the eukaryotes is the disassembly of all that tertiary structure. We have the DNA that is going around those proteins, those histones, they are positively charged. Now, if you need to replicate the DNA, you have to get it unstuck from those positively charged histones. So how are you going to do that? First thing you do is you modify the histones. You make it less positive. You put it a little bit of a negative charge, make it neutral. So it's not going to be so stuck to the DNA. It's going to loosen up a little bit. Now the polymerases can go in there and start replicating. But if you have this tightly coiled DNA and supercoiled and coiled again, it's basically impossible to replicate it because there is no space for the polymerase to get in there. So this is one of the main differences, because in the prokaryotes you don't have so many proteins that are interacting with the DNA. You have a more loose system.
C is for clue, that's okay with me.
So now what happens when you are modifying these histones? This is a very important thing to know here. In order to modify the histones, you have to go into the protein and change its properties. So what you do is that you add an acetyl group. You do acetylation of the histones. Now, what is this acetyl group? This is an acetyl group. Okay, so you have here this acetyl group that is going, these acetylases, they're going to get to the histones, add this group in there and make it less positively charged. It's going to make it neutral. So now you can start to unwind the DNA out of it, and then you can start your polymerization, your replication. But then after, when you finish the replication, you want the DNA to be associated with the histones again. So after you're done with the replication, the histones, you have newly generated histones without the acetyl group. Because remember, you are replicating the DNA, you're getting 2 from what you have 1 before, right? You have 2 chromosomes now and you had 1 before. You need new protein, you need new histones to associate with your new DNA. So you have a lot of histone production during that time as well. That histone is going to associate with the DNA, and then you're going to start calling up again. Now, another big difference between eukaryotes and prokaryotes. Prokaryote, circular DNA. Eukaryote, linear DNA. You have the ends, you have the telomeres. Now, there is a problem that happens here at the end of the DNA. You have here, especially at the lagging strand, okay, at the end of the lagging strand, you have a problem that, remember, polymerase has to add to a free 3' OH group. And right at the end here, there is nothing to add the polymers to. There is nothing to add the nucleotides here too. There is no free 3' group in there, right there. It's empty. So what's going to happen here? Polymerase is not going to be able to go there and synthesize that little piece of DNA that used to be the RNA, right? Well, most of the cells, which means the somatic cells, can't do much about it. They'll just have shorter DNA every time they divide. So if you keep on shortening the DNA every time you divide, eventually you're going to hit some important genes and you're going to die, because there is not going to be any chromosome left. All the chromosome is going to be shortening, and it disappears. So there must be something that the cell can do in order to prevent that. Now, there are 2 solutions to this problem. One of them involves this enzyme that is called the telomerase. And actually, this enzyme is only active in the gametes, in the embryos, and in stem cells. So if you think about it, the polymerase is the end of the chromosomes in these 3 cell types. They will never shorten. Every time that the cell is going to divide, especially think about it during embryonic development, you have a very massive amount of cell replication. You have a massive amount of cell division and DNA replication and synthesis of everything in there. So if you are going to cut out pieces of DNA every time you divide a cell, this embryo is already going to be compromised. That can happen. With these telomerases at the end of the telomeres on the chromosomes, these telomerases, what they do, they recognize the site at the end, and they will just stretch it out a little bit more in order to avoid the DNA from getting shorter. You're going to see more here. You add a repeat sequence, which you don't have to know the sequence, but just remember that you add a little piece of DNA to that end where it had nothing else to bind to. This is telomerase right there. The one more cell type that it is active is in cancer cells. So these cells really don't want to die. If they divide a lot and they keep on shortening the DNA, it's going to come to the point that they are going to die. They cannot sustain so many cell divisions because, you know, the tumor will keep on growing and growing and growing. The cells dividing. After a while, it's self-destructive. The cancer figured out that it cannot self-destruct, so it activates the telomerase gene. So you're going to find active telomerase in cancer. Now, this is also a good idea for people who are doing cancer research to figure out what can deactivate the telomerase. So now, instead of having a drug that will kill every single dividing cell in your body, you can have a targeted drug that, yeah, okay, it's going to kill the eggs and the sperm, but it will also kill the cancer. See, oh, this is a very cool video that shows exactly how the telomerase is working. But they did not have a video in English. But at least it's in Portuguese. I'll translate to you. Okay, now telomerase here, what it says, which is also a lot of detail, is that it's located in chromosome number 5. So this, what they're representing here, is just the chromosome and the site on the chromosome that you have the gene for the telomerase, which, you know, cancer cells, very clever, decided to activate it. So here it's showing that when you have the end of the chromosome, the end of the synthesis, telomerase gets in there. Let me make this bigger. Look at that. This is really neat. Telomerase gets in there and will just add nucleotides and shift to the end and add more nucleotides. Look at that. Now you don't really have a quite end to this. You see how it's elongating? The end here, this is supposed, oh, come on. This is supposed to be here, the end of that chromosome, the end of the DNA, that you have nothing else to add to. Now this telomerase gets in there, it recognizes this single-stranded end of the DNA. Remember that the telomeres at the very end of the DNA, and just stretch out this end. So now, when RNA primase gets in there, makes the RNA primer, you can get polymerase to come all the way to the end here and make the new strand of polymer again, of the DNA polymer again. You get the double strand again. Instead of having the DNA that ends up here, now you're still going to have a little part that is not synthesized, that is not duplicated, that don't have a double helix at the end, but you're going to start your double helix right here instead of right there. You gain your telomere back. The end is single-stranded, yes, it's still single-stranded, but now if you keep on increasing that end, so you don't shorten up your DNA size. Okay, so now it has to show you that it has to do with the gonads and also cancer cells. So basically this video is saying that if you can activate the telomerase on your body, you can basically make every cell of your body immortal, because when you shorten the telomeres, you are going to age. The cell ages as you shorten the telomeres. Everybody saw that that blog about the cloning the pet thing, right? That you have these people that are so crazy about their pets that they got the cell before the pet died, maybe a 15-year-old dog, and sent it to Korea and got the pet cloned. They inserted the pet DNA into the egg of another dog and came up with a puppy. The problem is that the DNA that was sent to this new egg was a DNA that was already old from a somatic cell of this dog. So it's going to be generated as a 15-year-old puppy. Okay, it's going to be born with every cell being already 15 years old, in biologically speaking, time. So this is why it's not such a good idea about the cloning. And I'm sure that the scientists would never quite disclose that little detail, you know. Oh yeah, yeah. They at least develop a lot of old age problems at a very early age. Yeah, like the dolly ship. But also, if you make all the cell immortalized, you could theoretically live forever, or at least, you know, maybe 300 years or so. And when I hear the stories, the biblical stories about Methuselah, right? They said he was about 300 years old. Maybe he had a mutation that every single cell in his body activated the telomerase. We don't know, but that would be one of the evidences, right? Living a long life. The only problem is that at the same time that you are regenerating every single cell that is good in your body, a mistake is going to happen sooner or later. If you keep on living a long time, chances are the mistake is really going to happen. There is nothing to avoid it. So at the same time that you are regenerating the good cells, you're going to come up with one cancer cell that's going to be regenerated and regenerated and regenerated. It's going to keep on growing. It's going to kill the rest of the organism. That's what cancer does. So it's a double-edged sword. Now, also, what is important to know here, well, the end of the chromosomes are not really garbage repeat sequences. They are just sequences that are there without meaning for a gene. It's more or less like those jumping genes ideas. You think they are garbage sequences? I'm sure they're there for a reason. So the end of these telomeres are also there for a reason. One reason is a very much of a kamikaze suicidal mission. It knows it's going to be cut off. Okay, it is going in there nicely and happy. It's at the telomeres at the end. It's a lot of repeat sequences of DNA that will never be transcribed. It's right at the tip of the DNA, and it knows it's going to be killed. It's going to be shortened and shortened and shortened. And as you age, as I mentioned, the telomeres get shorter. So at one point, if you lose a lot of the telomere sequence, you're really going to hit a gene sooner or later. And if it is a vital gene, you know, the cell goes. And also at the end of this telomere, when you have the single-stranded DNA, this is a very important site for interaction with proteins. Remember, if the DNA is naked, it's going to get killed. And there are a lot of endonucleases or a lot of digestive enzymes in the cell that will just love to find a single-stranded end of a DNA, because you just go there and chop it up so quickly and start killing that whole chromosome from the ends. The thing will disappear very, very quickly. Like the same way that you have a very high level of instability when you're working with RNA that is single-stranded. Well, one of the reasons is that you have a lot of nucleases or RNases that chop up the single strand. If you have free loading, the free DNA, I'm single-stranded here, totally naked, it's going to die. Okay? So instead of letting it die, the cell makes it associate with stabilizing proteins right here. That will go to the telomere and just pack it up with this shield so it doesn't get killed. Oh yeah, another thing that is very important, if you have loose ends of chromosomes, and if you have all this soup of chromosomes that are mixed together when the cell is ready to divide, chances are these chromosomes are going to snap back together. Remember when you're talking about the cloning in the class, in the lab? That if you have a certain endonuclease that has, like, for example, a blunt end cut, and you have these two chromosomes, these two pieces of DNA that are floating in there, it can just snap back together and connect to itself. So it's not the end of a chromosome anymore, now it's the whole thing. This cannot happen when you are doing DNA replication. Also, as I mentioned, this is very important for aging. And there are, I'm not quite sure if there are cases of progeria that are due to telomerase shortening. And I'm not sure which one came first, the chicken or the egg. Does the telomerase shortening happen because the child has progeria, or because the child has progeria, it cuts off on the telomeres? So I don't really know what happens first in there, but you can associate the old age or aged cells with shorter telomeres.